thanks for the introduction. My name is Andrei. I'm a student at Professor Wilhelm Huck Lab at the Radboud University in the Netherlands. And today I want to discuss a bit about what we've been doing with JCVI-syn3A and how we are using that as a model for building a minimal synthetic cell. So I will not talk much about our recent development of the syn3A cell-free expression system, but I'm going to talk about how we are using the system now. So initially when we talked with John and established this collaboration with Kate as well, our idea was to build a synthetic syn3A cell using the building blocks or the smaller parts of a living syn3A cell. So for example, we would isolate the cytoplasm, we would isolate the genomic DNA and the lipids, and putting those parts back together in vitro, we would like to recreate a synthetic version of the syn3A cell. Just to illustrate this idea, so you can imagine that you have a clock, and then you disassemble the clock, or you just break this clock into smaller parts, and then you put them back together in an orderly fashion, so in the end you have a functional clock. So that's the idea that we would like to perform with the syn3A as a model to build this synthetic version of syn3A cell. This approach seems can sound a bit simplistic in terms of experiments, but we believe that this might allow us to learn about the design principles behind building synthetic cells from the bottom up. This approach has been already used by some groups, so I'll give you one example here. By Professor Vincent Noirot from the University of Minnesota. He was able to reconstitute phages, viruses, from its genome. And the way that he did it, it was by isolating phage T4 genome and adding that genome into an E. coli cell-free expression system. And then at the end, he observed the production of fully infectious phage T4. So it's also important to note the size of the phage T4 genome is about 170 kb and with 289 genes. So our idea is to make some similar protocol, but using syn3A genomes instead of these phage T4. So our idea is to isolate syn3A genomes from living cells. Then we add those isolated genomes into syn3A cell-free expression systems. We have to encapsulate those into lipid-bound compartments, and then in order to recreate a synthetic syn3A cell. There are a few complications on doing this experimental protocol. The first one is because syn3A genome, despite this minimal size, is still larger than phage T4, for example, is about a twofold bigger than the T4 phage. Another complication is that we also have to encapsulate all this genomic DNA with the cell-free expression system into liposomes and make that system stable. And another complication is that our current cell-free expression system based on syn3A is not as efficient as E. coli, for example. So keeping those constraints in mind, we planned our experiments in three main goals. The first one would be building the functional syn3A as cell-free expression system. And then we would isolate intact genomic DNA and also optimize the expression of these intact genomes in vitro. And finally, encapsulate the cell-free expression systems with genomic DNA inside the liposomes, and also optimize expression inside the liposomes. So for the first goal, we published a few months ago already a paper in which we describe how we accomplish the cell-free expression system based on syn3A. Initially, we thought it would be very straightforward to just lyse the cells and just make the lysates and the reaction would go on. But we faced a lot of problems regarding the ribonuclease activity present in the mycoplasmas in general. And the way that we succeeded that is by using a very specific cell disruption method called nitrogen decompression. It's here in the left side. In this technique, you incubate the cells at a very high nitrogen pressure, which allows the nitrogen to dissolve inside the cells. And by instant decompression of the cells, they disrupt from the inside out. In this way, we got a cell-free expression system based on syn3A, which was functional. And together with the feeding buffer and also with tRNAs that were isolated by Evan, that was a very important component in the system. We were able to detect the protein expression, protein production, using this plasmid coding for a reporter protein. In this case is M neon green. So in the right side, you can see the graph in which we show the protein expression over time. And it was either controlled by an exogenous T7 RNA polymerase or by endogenous RNA polymerases, which were already present in the lysate. So with this, we accomplished the first goal, which was having this syn3A CFE system so we can use for other purposes. So next, we wanted to isolate genomic DNA from syn3A and optimize the expression of these isolated genomes in vitro. In the previous years, we also performed some similar experiments, but using E. coli. So it serves as a proof of concept here. So what we did is that we designed a E. coli strain, which has a GFP gene in the chromosome. We isolated these E. coli genomes and we expressed in vitro. So you can see the expression of GFP here over time. Those are just bulk reactions. And we were also able to encapsulate this E. coli genomic DNA inside the liposomes together with the E. coli CFE system. In this case, we used a capillary microfluidic system to make the liposomes. And we saw expression of GFP over time here up to 15 hours. Those experiments were just a proof of concept using E. coli, but what we are currently working on is similar experiments, but using syn3A genome instead. Which brings me to the conclusions of this presentation. Now we have these functional syn3A systems which can use to reconstitute this entire syn3A cell. This can be a bit more complex compared to the phage experiments that I showed in the beginning in terms of the size of the genome, the requirement for compartmentalization, and also the efficiency of the cell-free expression systems. I showed also this proof of concept that we can indeed isolate intact genomic DNA from bacterial cells, and we can also express genomic DNA inside the liposomes. And probably for the future, once these systems are working more or less efficiently, of course, we're going to have to look ahead into the synthetic cell itself and think about expression of several genes at the same time, because here we're just thinking about expression of a single reporter gene. But also we think that the syn3A CFE system can be used for several other experiments to study the minimal metabolism and study the gene expression in vitro. And here I just put the QR code for our recent paper in ACS Synthetic Biology. So if you are curious to see how did we succeeded to make this functional syn3A CFE system, you can use this link. And I'd just like to also thank our collaborators, our technical staff in the university, and also our collaborators in North America and South America, and BASIC for funding our project. Thank you, and I will be happy to take any questions.
Anyone before I start? Andrei, so I think it would be good if you can initiate. So listen at the end of today's session to the Sayans group from Dresden, and they can give you will get some better ideas about how to construct a mycoplasma-like lipid vesicle, which may not be important, but it might be absolutely critical.
Yeah.
How do you think we're in the ballpark with sufficiently active cell-free systems?
Yeah, that's a good question. So the expression in general is quite low if you compare to E. coli, for example. But expression in general, we achieved about 300 to 500 nanomolar, which is not bad if you compare to the concentration of single proteins in the cytosol, not the total protein. But of course, there's a problem about efficiency. And if we think about expressing several genes at the same time, there will be a problem of resources as well. And yeah, I think there's still a bit of a room of improvement in terms of efficiency of the transcription translation that needs to be solved as well.
How large are the vesicles that you envision making relative to the size of these natural cells?
Yeah, so that's a bit of a limitation of the method that you choose to make these liposomes. I would say that it would be in the micron scale. I think the smallest that you can go for is like 5 microns diameter. So those ones that I showed in this slide, it was about 50 microns. So that's quite larger than the cell itself. So
and then how do you plan to get, so I worry about the process of putting a genome into a vesicle. How intact will it be? Now, an E. coli genome is roughly, 8 times the size of a SYN3A genome. Talk about that, please.
yeah, so I think the larger the genome is, the more fragile it is for encapsulating, especially if you are using microfluidic systems that you can have lots of shear forces during the encapsulation. So the smaller the gene is, I think that's the best. So I think we are at some point some advantage of working with the SYN3A genome because of the smaller size compared to E. coli. So, but we have to do some studies yet about the shearing forces during the encapsulation. We didn't check, like after the encapsulation, how intact is the genome itself. Yeah, that's a very good question.
We should have a conversation soon.
Yeah.
Other questions for Andrei? Come on, folks. Pay attention. More questions. You guys need to be doing better.
I have one then.
Excellent.
Which is, so I think one of the challenges about encapsulation is membrane proteins and what happens with those, and can vesicles function like membranes if they don't have the membrane proteins? So I'm wondering if there have been discussions at all about trying to do some sort of membrane prep and then create vesicles from cell membranes that would have proteins still in the membrane. So essentially ghost membranes in a way.
Yeah, that's a very good question. We don't plan to do that yet. We are just thinking about pure lipids membranes. But definitely we should think about adding membrane proteins to the membrane. But it's not in the plan yet.
Okay, because I envision that being one of the sticking points that you'll have to overcome at some point.
Thank you.